Alright, so back to the retina. So with the retina, we have the macula lutea, and we also have something called the optic disc, and this is our blind spot in our vision. And if you have the textbook, and I think in both all textbooks have this optic blind spot test, but the interesting thing is that your retina isn't completely in, it's not all the way, or it's not a complete layer. There is a little spot, and why do you have that optic disc in the spot? Well, basically you have all of these nerve fibers and these cells running this way. So where they all kind of gather together to form a bunch of like, yeah, all these optic nerves and all these extensions of these axons enter past or go past the eyeball and enter the optic nerve. This is the blind spot. So there's no retina here because why? This is all occupied by nerves. The nerves need some way to exit the eye. If you want to, I mean, cool thing is that some organisms, like I think octopuses or octopi, they don't have blind spots because they have a different arrangement of these nerves, but we have a blind spot. And that's what we see here. So not only we have nerves, but we also have all these blood vessels as well. So nerves and blood vessels need a way to enter and exit the eye. So this is why you see all the vasculature and all the nerves entering the eye over there. Now this macula over here is what we call, so macula is important because it contains a very important structure called the fovea, but there's also a condition called macular degeneration, and it's pretty scary because this is the center of the focus of our eye. So if you have the cells in retina start to degrade and all this, the cells of your retina start to are unhealthy and they're not intact, then you can end up with this. So the macula is very important because this is our focus of our eye. This is the highest detail area. So these people who have macular degeneration, they have vision, but imagine trying to read something where you can't center your eyes on it and you kind of have to like move your eyes around so you can kind of see things. So people who have this, they have a visual impairment. Now, in the middle of the macula, you have something called the central fovea, or sometimes called fovea. So the fovea is a little pit. It's actually a little indentation, and you can see that right here. And the cool thing is that it contains all these things called cones. And let's talk about rods and cones. So it's a central fovea is a tiny indentation in macula lutea. It only contains cones, and this has the highest visual resolution. Thing about your retina is that it actually has like many different parts of, I mean, even though it has visual cells everywhere, the the central fovea is like the 4K of your vision. Like again, things in your peripheral vision, you can see them, but they're not as clear as things you see with your central fovea or when you're reading or looking at this presentation. You have in the center, that's where you have higher resolution. But for your peripheral vision, this is why you don't have as much resolution. So the cool thing is that this is why when you're reading, if you look at someone reading, this is why you see their eyes kind of darting like that all around and bouncing around, is that they're trying to focus on the details and these little small movements. Now, this is, I think, a cool example. You can do this at home, is that there are 12 dots in this image, and look at the slides, and your brain won't let you see them all at once. Well, it's not so much your brain, but the thing is that with these small dots that are very detailed, when you're focusing on one dot, you're kind of focusing your central fovea on it. But everything else around it, the further it is from that central fovea, is going to be kind of hazier and less resolution than the part that your eyes are focused in on that. So yeah, it involves your brain perceiving that, but being able to focus things. This is why you're not able to see all 12 at once, unless you take a very big step back. But if you're looking at it like right up close, this is why you can't see all of them at once. And then we have the retina, and then with cones and rods. So yeah, this is why your central fovea, and that's why macular degeneration is I mean, it really severely affects someone's quality of life. Yeah. So here we have multiple things, but there's many types of cells, and the ones I definitely want you to focus on are cones and rods. So there are many things, and yeah, it's kind of cool. You have melanin in the epithelium of your retina as well, and it helps to kind of get absorbed. So if the thing is that you have light entering your eye, but you want it bouncing around all of the inside your eye, so it's kind of like a black background that helps to absorb light. But you also have these cells, very important cells called rods and cones. So rods are basically kind of like detecting your sense of light and dark. And then cones over here, these are important for color vision. So cones and rods, it's only showing one cone, but there's actually multiple types of cones. But what we have with rods, now this is a little molecular stuff, but what I want to show you here is that this is a protein called opsin, and if you are taking like a molecular biology class, you think when you have this and it's this retinol, this chemical compound right here, rhodopsin is what we call a G protein coupled receptor. But again, that's more for those of you going to medicine or biology or molecular biology. But what's important here is that retinol is actually derived from vitamin A and carotenoids. So this is why it's very important if you're a nutrition major, this is your moment. This is why it's very important to get carotenoids in your diet, because if you don't get carotenoids and won't be able to make retinol, and if you don't have retinol, you won't be able to make these compounds that are needed for your eyes and these rods and cones to properly function. So this is why you need that vitamin A. And the cool thing is that, there's wavelengths of light, and wavelength basically refers to how short these waves are. What I'm getting at here is that rods, blue, green cones, they detect different colors of light, and colors of light are determined by their wavelength. If you want to know more about wavelength, I guess we do cover it when we talk about the hearing and vision, and hearing. But why is that important? Because color vision involves light and interpreting. So this is why there's 3 types of cones. They detect blue, green, and red. And you might be like, wait, it's like when I was in elementary school, I learned like the colors of the rainbow. Well, you make all of them by mixing red, yellow, and blue. Well, color vision and mixing light is different from actual pigment and art. So with mixing light, you have red and blue, but instead of yellow, you have green. So red and blue, they form this bright pinkish, purplish color called magenta. And if you know computer science, if you've done graphic arts, you already know this stuff. But instead of yellow light, again, we have green light, we have green receptors, so green and blue together, they make this light blue, this aqua color called cyan. And then what about red and green? Well, this is, again, mixing light is different than mixing paint. So when you mix red and green, like if you have red and green paint in elementary school, you know it makes brown. But when you're mixing red and green light, you make yellow. And then when you mix all 3 together, and if you remember all the way back to art class where you mix all the paints together and you made this weird kind of grayish brown color, well, if you mix red, green, and blue, then you get white. So the big thing, again, if you've done graphic arts or anything computer related, this is why you have that color wheel. It's different than the color wheel you learned in art class and painting. So my big thing is that mixing colors of light is different from mixing colors of paint. But you're able to get the entire spectrum of this, and this is why we have that 4K and all that. If you actually look at a computer screen or that 4K screen, they're all red, green, and blue little small lights, and just to a small resolution that we're not seeing the individual ones. But by adjusting the levels of red, green, and blue, we can affect the different levels of red, green, and blue cone activity in our eyes, thus giving ourselves color vision.